In this video, we're going to talk about the 10 actual steps of glycolysis. So things I want you to keep in mind. I want you to pay attention to the names of the enzymes that I write down, and because they generally make sense. Like the name of the enzyme will help you understand what the enzyme does and what it acts on. In addition, I want you to pay attention from step to step. What happens to the molecule? What part of the molecule changed? That's also going to help you remember what's going on. In addition, I want you to note in particular that there are some reactions that are written with a single arrow and some that are written with a double arrow. The ones that are written with single arrows can only the enzymes that have only a single arrow only catalyze that forward reaction, whereas the enzymes with double arrows can catalyze both the forward and the reverse reaction. So let's hop right to it. So as we said earlier in the previous video, glycolysis is the splitting of sugar. So we start off with glucose, which is a 6-carbon molecule. So this is glucose, alpha D glucose to be specific. So what we're going to do is we're going to add an enzyme called hexokinase. Now what is hexokinase? Hexo, or hex, is the root for 6, right? So if we numbered these carbons here, this would be carbon number 1, 2, 3, 4, 5, and 6. And kinases add phosphate groups from ATP, right? So we would expect hexokinase to add a phosphate group to the 6-hexo, the 6 carbons OH. So we're going to invest an ATP into this reaction. We're going to invest an ATP because and then that phosphate from that ATP will be attached to the 6th carbon. So then the only difference between this molecule is alpha D glucose and this molecule here is that we've attached the phosphate group to this 6th carbon up here. So the ATP becomes an ADP. So we've invested energy to start off this process. So hexokinase, the enzyme, makes perfect sense. It's a kinase that adds a phosphate group from ATP on the 6th carbon. And then we get this molecule called glucose 6-phosphate, which is just glucose with a phosphate group on its 6th carbon. Nothing too complicated about that. The next step is catalyzed by phosphoglucose isomerase. So it's an isomerase. Now isomers have the same molecular formula, so we know that nothing is going to be added or removed from this glucose 6-phosphate. So all we're going to do is isomerize it. We're going to move it around, change the structure of it. It's an isomerase for a phosphoglucose. Well, this glucose has a phosphate on it, so it's a phosphoglucose isomerase. It's going to isomerize the phosphoglucose, specifically glucose 6-phosphate, and we end up getting this molecule here, which is fructose 6-phosphate, which is still a 6-carbon molecule, and the carbon is, or the phosphate group is still in the 6th carbon over here. But since it's an isomerase, nothing went into or out of this reaction other than this particular substrate. Cool. Now the next step, we're going to take fructose 6-phosphate and we're going to add the enzyme called phosphofructokinase 1, also known as PFK1. Okay, so what is this? Phosphofructokinase. Kinase. It's going to add a phosphate group from ATP. Okay, where is it going to add it on? It's going to add it onto the phosphofructose, which is fructose with a phosphate on it. So this enzyme will act on the phosphofructose, which is fructose 6-phosphate, and it's going to add a phosphate group from ATP onto the 1 carbon. That's why this 1 is here. So we're going to have an ATP invested so that we add a phosphate group onto this 1 carbon. So the difference between this molecule and this molecule is that we've added a phosphate group here. So what's left of that ATP is an ADP. So this is the second time. So here's our first investment of energy. Here we just invested our an energy the second time. One thing I'd like to note is that this particular step is the first committed step of glycolysis. Okay, which I'll elaborate on later. First, this is kind of small, sorry. The first committed step of glycolysis. So this enzyme is going to be definitely highly regulated. Again, single arrow. So is hexokinase. We'll talk about that later. But the whole idea is that this is the first committed step of glycolysis. So it's going to be a highly regulated enzyme, and we'll see that later. So what do we create here? Well, we added a phosphate group onto this one carbon here, onto this to the fructose 6-phosphate to create this molecule here, which is fructose 1,6-bisphosphate. Does that name make sense? Well, this is a fructose, so that part makes sense. Bisphosphate, it has 2 phosphates. Where are the phosphates? On the 1 carbon and the 6th carbon. So let me number this really quickly to make sure that makes sense to you. 1, 2, 3, 4, 5, 6 carbons. Okay, so fructose 1,6-bisphosphate. Again, that name makes sense. So is that an enzyme name? Okay. Next step, the 4th step of glycolysis. So at this point, what we're going to do is once we've created fructose 1,6-bisphosphate, this thing has committed to glycolysis. So what we're going to do here is the enzyme aldolase. I don't have a fun or sort of meaningful way to remember aldolase. This one's kind of just remember this one. But this one, aldolase, is the enzyme that cleaves fructose 6-bisphosphate, the 6-carbon molecule, into 2 3-carbon molecules. So those 2 3-carbon molecules are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Let's see if these two names make sense. Dihydroxyacetone. So acetone is a ketone, right? A ketone. This here's a carbonyl group with 2 carbons on either side of it. Now, if this was a CH3 and if this was a CH3, then it would just be acetone. But this is a dihydroxyacetone. So instead of a methyl group here, we have a hydroxyl group here, a CH2OH. Okay, but it says dihydroxy, so we would expect this to be a CH2OH. But it's not, right? Instead, it's a phosphate. So this is dihydroxyacetone phosphate. That's why this thing is here. If this phosphate wasn't here, it would be dihydroxyacetone. Okay, so again, this name makes sense if you think about it. This here is glyceraldehyde 3-phosphate. So we would expect to see a glyceraldehyde and a phosphate on the 3rd carbon. So the aldehyde would be the number 1 carbon. So this is CHO, right? That's the aldehyde up there. Glyceraldehyde comes from sort of the glycerol, so we would expect that the 2nd carbon to be a CHOH. And this last one, if it was glycerol, it would be a CH2OH, but it's not a CH2OH because the phosphate group's on the 3rd carbon. So we have the CH2O and then the phosphate group there. Okay, so the 2 phosphate groups from the fructose 1,6-bisphosphate are here. One's on this carbon, one's on this molecule, and one's on this molecule. Now, what are we going to think about? Something very important happens here. Now, between these 2 molecules, glyceraldehyde 3-phosphate, abbreviated as G3P, and dihydroxyacetone phosphate, abbreviated as DHAP, only one of them can go on further in glycolysis, and that's glyceraldehyde 3-phosphate. So what we need to do is convert the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So the enzyme that catalyzes that reaction, the 5th step of glycolysis, is called triose phosphate isomerase. So triose phosphate isomerase. Triose means 3-carbon sugar. Phosphate means that 3-carbon sugar has a phosphate group on it, and it's going to isomerize it, which means it's not going to add or remove anything. It's just going to move around the atoms that are already there. So this is the triose phosphate. It's a 3-carbon molecule, with a phosphate group on it, and we're going to isomerize it. We're just going to turn it into G3P. Now, after this 5th step, we have 2 G3Ps, which is why I do this pink bracket here. That means every single step that happens here on out happens twice, once for each glyceraldehyde 3-phosphate. So everything here on out, from here onward, everything happens 2 times. Okay. So once we have glyceraldehyde 3-phosphate, what it's going to do, we're going to put an enzyme in here called glyceraldehyde. The 6th step is glyceraldehyde 3-phosphate dehydrogenase. Okay. If you recall, I mentioned that you would associate excuse me, the term dehydrogenase with redox reactions. So something was oxidized and something was reduced in this case. That's what we're gonna know. And when we think about redox, we want to think about NAD plus NADH or FAD FADH2. In this case, we're going to be thinking about NAD plus or NADH. So what happened here? Well, if we notice on the third carbon here and the third carbon here, nothing changed. We still had a CH2O and then the phosphate group. So nothing changed on the third carbon. The second carbon, CHOH, CHOH, nothing changed there. Here, we had a CHO, right? Here, now we have a COOPO32 negative. Okay, so what happened? Was glycerol di3-phosphate oxidized or reduced? Well, here it had this carbon was bound to this carbon, and then a single bond to hydrogen and a double bond to oxygen. And just to sort of show you that this would actually look like this, right? It's an aldehyde, right? It looks like that. So this carbon had a bond to carbon, a single bond to hydrogen, and 2 bonds to oxygen. Now it still has that bond to carbon, still has that double bond to oxygen, but now instead of a bond to hydrogen, it has a bond to oxygen. So glycerol di3-phosphate was oxidized to 1,3-bisphosphoglycerate. Okay, so if this was oxidized, something must have been reduced. Well, the thing that was reduced was NAD+. So we went from an NAD+ and created an NADH. So what really happened? Glycerol di3-phosphate was oxidized, NAD+ was reduced to NADH. In addition, notice that we added that phosphate group, right? Where did that phosphate group come from? That phosphate group came from an inorganic phosphate from the environment. Okay, so that's going to be noted as P sub I. Okay, does this name make sense? 1,3-bisphosphoglycerate. Well, the phosphates are on the 1 carbon and the 3 carbon, so 1,3-bisphospho, 2 phosphate groups, glycerate. Looks very much like the next molecule, which is actually 3-phosphoglycerate. Okay, so the 7th step is catalyzed by phosphoglycerate kinase. Actually, before I elaborate on this one, I wanted to make sure that this makes sense. Glycerol di3-phosphate dehydrogenase. The dehydrogenase, the redox reaction, occurred on the glycerol aldehyde 3-phosphate. In case that wasn't perfectly clear. Okay, so phosphoglycerate kinase. This enzyme is kind of a little bit weird. We associated kinases with adding phosphates to things. What actually happens here? I remember if this is why the introduction video to glycolysis is important. At this point, we've already invested 2 ATPs. So now we know we're not going to be using up any more. We should only be making some. So in this case, what's going to happen? Notice what happened to the molecules. Carbons 2 and 3, CHOH, CH2, and then this phosphate group. Over here, it looks identical, carbons 2 and 3. The change occurred at the first carbon. Here, we had the phosphate group. Here, we no longer have it. We just have this carboxyl group. So we lost that phosphate group. Where did that phosphate group go? Well, it went onto an ADP to create an ATP. So here we made an ATP. Okay, so this phosphate comes over onto this ADP to make ATP. Okay, and then what we're left with is this 3-phosphoglycerate. So now we only have one phosphate group left here. Okay, now why is this called a kinase? Why is this called phosphoglycerate kinase? Why does that make sense? Well, this enzyme was probably named for the reverse reaction. If you think about the reverse reaction going from 3-phosphoglycerate to 1,3-bisphosphoglycerate, you'd be taking an ATP and going to ADP, and that phosphate would be attached here to make this. Right, so this enzyme was likely named for the reverse reaction, maybe because biochemists discovered that reverse reaction before the forward reaction. Okay, so next up, step 8. What happens here? We're going from 3-phosphoglycerate to 2-phosphoglycerate. So the only thing that happened was we took the phosphate group from the third carbon and moved it over to the second carbon, and then so the change occurred there. So we didn't add or remove any atoms. So this is kind of like an isomerase. So it's actually not an isomerase, unfortunately. That's kind of confusing. But instead, it's phosphoglycerate mutase, which, I mean, still makes some sort of sense. We're mutating it. Whoops. It looks a little bit different, but that's the name of the enzyme. As far as why, I don't know. Maybe scientists just wanted to confuse students like us. Whatever it is. But the point is, it is sort of like an isomerase in that all it does, it just moves the atoms around, doesn't actually add or remove anything. Okay, so then step 9, what we have is we're going from 2-phosphoglycerate to phosphoenolpyruvate. So what's the difference there? The one carbon, nothing happened. We still have a carboxyl group up there. Here, what happens is we still have this phosphate group there, but we end up forming this alkene here. So now, the enzyme that catalyzes this step is called enolase. If you recall from organic chemistry, an enol was essentially an alkene right next to a hydroxyl group. So here, what we're doing, we're making phosphoenolpyruvate. Notice how this looks. This is an alkene next to what would be a hydroxyl group, but instead this is a phosphate. If this was an alkene and this was an OH here, that would be an enol. But this is a phosphoenol. So there's the alkene and then this phosphate group instead of the hydroxyl group. How did that form? Well, what ends up happening is we lose this hydrogen and this hydroxyl group. We lose them both in the form of water to form that double bond there. Okay, so we lose this H, we lose this OH, and then we form that double bond there, and then we're left with phosphoenolpyruvate, which is a very high energy molecule. And when it's converted into pyruvate via the enzyme pyruvate kinase, oh goodness, I'm moving on to this and I haven't mentioned enolase. Enolase makes sense, right? It's the enzyme that forms this phosphoenol. Okay. Phosphoenolpyruvate then is converted into pyruvate. So what's the difference between these two molecules? Well, the carboxyl group stays the same. The methyl group, we form the methyl group, right? We get rid of this alkene, we get rid of this phosphate group. Where does that phosphate group go? Actually, before I even mention anything, at this point, how many ATPs have we invested? Well, we invested 2 up here. Let me scroll back up for a second. We invested 2. We invested 1 here and 1 here. How many ATPs have we made so far? 1? No. We've made 2, right? Each reaction here creates 1 ATP. But recall that we have a glyceraldehyde 3-phosphate. There's 2 of them, right? So each reaction after this happens twice. So we've created at this point 2 ATP, and we've created 2 NADHs. So at step 7, we've already paid back our debt. Okay, so now here, this is the reaction that was the final reaction. So we're actually going to net our ATP here. So this phosphate, where does it go? It gets tacked on to ADP to make an ATP. Okay, notice I have a little green star here to indicate that this enzyme is allosterically regulated. So here we have pyruvate. We specifically have 2 pyruvates. Okay, so if you recall, the introduction video mentioned the products. So hopefully this all makes sense now, how we got all that. So let's do a quick little recap of what we covered. The steps above that have the little green star next to them are the allosterically regulated enzymes, and the next video will be covering that. As far as the recap, the first reaction was the first energy investment. You can scroll back up, pause the video, and check that out. The third step was a second energy investment, and that was the first committed step, catalyzed by phosphofructokinase 1. That, if it's the first committed step, we know that first committed step must be allosterically regulated. At steps 4 and 5, right, we have to realize that only G3P, glyceraldehyde 3-phosphate, can go on in glycolysis and continue. So everything made after that point is made twice because there are 2 G3Ps. The 6th step is a redox reaction, right? Which is, we know that because of the dehydrogenase, and of course that's when the coenzyme NAD+ comes into play to be created into NADH. The 7th step is with the first substrate level phosphorylation. So substrate level phosphorylation is just this idea that here we're taking this enzyme, this phosphoglycerate kinase, is acting on this substrate, 1,3-bisphosphoglycerate, and it's taking that phosphate off of there and adding it onto ADP to make ATP. So that's what substrate level phosphorylation is, just taking a phosphate off of a substrate and tacking it onto ADP to make ATP. Okay, so that's the first substrate phosphorylation in that 7th step. At that point, we've paid back our 2 ATPs, right? Because that reaction happens twice. And the 10th step is our second substrate level phosphorylation, and that's of course when we net the 2 ATP. So overall we've created 4 ATPs, but we net 2 of them. It's because 2 of them have to pay back our initial debt from these 2 energy investments. In addition, just as a keynote, something I'd like to mention here is that the 10th step, going from phosphoenolpyruvate to pyruvate, there's a lot of energy released. And that energy release powers the production of ATP. But there's enough energy released to power the production of 2 ATPs. But only one is made, which is interesting. It's a little bit inefficient in terms of energy. Why doesn't that reaction power the production of 2 ATPs? It would be ideal if it could. So it's interesting. Maybe if you become a biochemistry researcher, and if you can, or a biochemical researcher, whatever, or a bioengineer, who knows? If you can somehow engineer a way to make it so that that particular reaction produces 2 ATPs instead of 1 ATP, I mean, that'd be pretty awesome. That would just make biological organisms much more energy efficient. So that'd be kind of cool. That'd be pretty interesting. So that's about it. Thanks for watching the video, guys.